final list of who's going to what group. And lab will also be done a bit differently this time. There will actually be 5 different experiments. It's actually to give everyone an idea of what it's like to do an experiment, an actual experiment from beginning to end. So, for example, groups 1 and 2 will work on one experiment, groups 3 and 4 will work on a different experiment, groups 5 and 6 on a different experiment, and so on. Part of what you will do during lab is also to take a video of what you're doing, so you can actually share it with your lab mates later. So in some ways, it's sort of like teaching your friends what you did, and most of the marks for lab will actually just come from there. So hopefully it will be a very simple, easy lab for you guys to work on, and it won't be so stressful because I know at the end of the semester it's quite stressful for you guys. Test will be held on 17th January. It's only just on one topic, synthetic biology. There's just 3 questions. It'll be sort of short essays. So you guys will just come here. We will hand out the actual test. You guys will work on it here and then just hand it back to me at the end of the lecture session. So test will be on 17 January, 4 to 5 p.m. here. Since the end of the last few classes sort of coincides with Chinese New Year, after talking to a couple of people, I think what I will do is I will try to combine the last 2 lectures into one lecture and talk as fast as possible. Unfortunately, I cannot have online classes, whether it's recording or face-to-face online, that's not allowed for biology school. So the suggestion was to combine the last 2 classes to 19th January. That way when everyone goes home for the holidays or Chinese New Year, you don't have to worry about coming back until the actual exams. If people are not okay with that, please let me know and we'll try to find something else that works. But again, the last class then should be on 19th January. Everyone okay with that so far? Okay, cool. And then the last one is, do you guys want a review session? Since it sounds like a lot of you won't be coming back, if you do want a review session, let me know when. The review session, since it's not actually part of the official class, can be held online. So if you guys do want a review session, again, just let me know when, whether you want it that week of 26th January, or you want it sort of closer to your exam, the actual date of the exam, let me know. I'll be happy to schedule sort of an online thing. Or if you guys don't have our review session and just want to just send me individual emails or messages, you can also do that as well. All right, any questions? Okay, all good. I'll also make this announcement in the group chat on an e-learning if anyone came in late. Right, hopefully everything's okay. So we will get started. Alright, we are down to, after this I think we have 3 more classes to go. So application and ethics of genetic engineering part 2. As usual, let's start with a review of everything we've done. So in our last class, we talked about what people used to do before genetic engineering or before genome editing actually came into, or before it was actually discovered. So a lot of people use conventional methods such as dietary restriction and supplementation. So these are things, for example, if you lacked vitamin C, you would just take vitamin C and try to fix it that way. A very special way of fixing things without actually addressing the core problem of what went wrong genetically or through the mechanism of it. We also learned about a couple of other therapeutic techniques. So even though today we talked about mostly solely CRISPR, but before CRISPR, especially in the 80s and 90s, there were things like pre-LOX recombinase. Hopefully everyone remembers how that works. So you have your LOX P site, and if you have your pre-enzyme, you're able to create conversions or deletions, for example. We also talked about sleeping beauty transposon. There are other transposons as well, but again, it's able to move certain sequences to a different region in your genome. We also talked about antisense, ribosome, and RNAi, all of which inhibits transcription and translation because of the complementary sequence to your gene or to our region of interest. And then finally, we talked about the genome editing tools, think human nucleases, talent, and finally CRISPR. Whenever we talk about genome editing as a whole, or actually anything that you've learned throughout this class, something that's really important is your delivery method. So if you do want to deliver it to your organism of interest, typically people would use viral methods. We did also learn other methods, especially in transgenic animals. But if you're just doing the work in a lab, so in vitro work, people also use non-viral methods. Finally, we talked about implications of genome editing. We watched a couple of clips. So I guess even from 20 years ago, the main concern whenever there's a new technology is what would happen if people actually apply this to humans. Will we actually get things like designer babies? And what is the implication of that on the population as a whole? Finally, we addressed, or we talked about certain hurdles and risks. So actually, how easy is it to actually come up with things like a designer baby? And there's actually so many different things that we have to know. So I guess in some way, it's not really as simple as what people think. Right, in today's class, we will go over just a brief overview of timeline of genetic engineering. Hopefully that will help you sort of remember or bring together all the different concepts that we've learned in the class so far, genetic engineering applications, and simplified physical workflow. Right. Whew. So when something that we touched in the class over and over again is we learned a lot about recombinant systems, and that's sort of related to gene splicing, which was actually done in 1971. So the ability to take different components or different genetic regions from different organisms and piece them together. So that was actually one of the key discoveries that actually played a huge role prior to genome engineering. And then in 1982 was when there was actually the first genetically engineered synthetic recombinant protein. So again, once you know you can make a recombinant protein, there's a lot of different things that you can do with that. 1996 was a big year because that was when the first commercial GM crops were planted in a very large area. So this was at the end of the 1990s. Since then, there's been lots of different fields of different crops, not just tomatoes, all around the world. And then finally, in 2012, sort of focusing on different components, primarily in bacteria, the first genome editing tool or CRISPR was discovered. We touched this in our last class, I believe. So we talked about the different applications, primarily in industrial biotech, that a lot of you might be involved in one day, but it is used extensively in microbial fermentation and therapeutics by production. It is used a lot, and in Malaysia, this is especially true in therapeutics and bio-research. So this is for gene and cell therapy, so actually trying to fix a certain disease or understand the mechanism of a certain disease through disease models and understanding biological pathways. Malaysia is sort of gearing towards genome editing in crop plants and farm animals. Even if you look this up in the world as well, people are using this for all sorts of plants and animals to address all sorts of things, not just for animal welfare, but also to make them to be able to adapt better to things like climate change, to make them more resilient to certain diseases, and also for the end use of this, so being able to eat something which is more nutritious or better for us. Right, so these are just some examples. There are obviously tons of different examples out there, but I just sort of listed some that have been quite interesting. So in crop plants, there have been modifications of larger grain size. So if you do that again, you have more nutritional content in a single grain. That requires modification of GS3 gene, increased protein content. They do this quite a lot. So not just increased protein content, but improved oleic acid content, improved digestibility in maize as well. So you make this sort of think of some of the different restrictions that people have in regards to food. Some people cannot take gluten, for example. Some people are allergic to eggs and nuts and shellfish. Some people are lactose intolerant, so people are actually able to use genetic engineering to sort of modify all that and come up with different flavors. For livestock, you have allergen-free milk. Honest dairy cattle, we saw this in one of our slides, cold tolerant livestock. This is a knock-in and disease-resistant animals. So all those in the brackets, those are examples of the genes that were modified. Great. So the question then is again, how easy is it to actually do that? So in the next couple of slides from now to the end of the lecture, we'll just be going over a simplified CRISPR workflow in plants and animals. So overall, and you'll be doing you already did number 1 and 2 in your last lab, and you'll be doing number 3 in your next lab. So overall, if you have to talk about this workflow, these are the 5 main components that you should pay attention to. Again, since you've already done 1 and 2 in your last lab, hopefully it's something that you can follow along easier. So whenever you want to make any modification, obviously you have to pick the genomic target. You have to know what you want to modify specifically. So even in this that we saw earlier in crops and livestock that's been modified, everything they only modified a single gene. So you have to know what that single gene is to give you that phenotype or that genotype that you want. So when we talk about genetic modification, if occasion of most of the time people do a genetic knockout. So you know that something is causing a disease. So if you knock out that gene, maybe you're able to prevent that disease from happening. So in cases like that, people tend to target the 5' region, and then if you target something in the 5' region as close as possible to the promoter, they're able to prevent transcription and transmission of that protein of interest. Occasionally, people just want to repress a gene, so perhaps at a certain time or activate a certain gene. So this was used, I believe, in I think it was thalassemia, I think, gene therapy. But it's something along the lines of, say, you want to repress a gene during a particular developmental stage, then you can do that. And then once it's passed as developmental stage, maybe you want it to be expressed again. So in cases like that, it's not a permanent removal of the gene or not a permanent reduction of the function of the gene, it's just temporary. So in those cases, you tend to target the promoter region. Occasionally, people also use it to alter protein functions. So in this case, they still want the protein to be there, but they just want to stop the protein from being active. So you can also target non-protein domains, or you can also introduce knock-ins or introduce a completely new gene itself. And that is usually as close as possible to your region of interest. Alright, so you guys already did this, so the next few slides should be very, very familiar to you guys. So whenever you want to do any CRISPR work, typically you start off with your gene of interest. I guess in our labs, we straight away went to the gene that was listed in the manual. But if you are actually doing this in real life, it's just a lot of reading papers to figure out what genes may be important. A lot of times it may be 3 or 4 different genes, but then you can sort of apply the knowledge that you learned from the synthetic biology classes. So how do you actually use an educated guess to figure out which gene is most important? So once you have that gene name, you can actually input it. It will give you all the information about your gene of interest, and then you can use that and proceed to your next step, which hopefully you remember from the lab, is actually design your guide RNA. So when it comes to the guide RNA, hopefully again, you guys remember, you guys looked at on-target and off-target sites, so this is something that came up a lot in discussion as well. On-target is the gene that you want to target specifically. So you want to have a cattle with no horns, so you want to be able to target the gene that codes for horns. You don't want it to target, you don't want it to have an off-target, so you don't want it to suddenly make, change the color of the cow to having blue color hair. So if you do something like that, that's off-target. So again, anything that you don't want is off-target. And to do all this, you have tons of different online softwares. This one I won't go over too much, but if you guys sort of go back and look at what the CRISPR-Cas9 system looks like, we talked a lot about the guide RNA and the vector, but something else that is important is known as a campsite. A campsite is a lot like traffic light, like a red traffic light that says, hey, stop here, and this is where you should target your gene of interest. So even if you go back and you look at your lab 3 results, the campsite is what determines where your guide RNA should be. In the lab report that you guys worked on, that one actually used NGG, the first one, and that's just related to which bacteria that your CRISPR-Cas9 come from. So different bacteria have this sort of different stoplights. So if you use things like Cas8, Cas9, you would need that N and G RRT sequence for you to actually tell you where your guide RNA would look. Everyone okay so far? Anyone needs me to repeat anything? Cool. Alright, so next you guys did this in your lab as well. I don't know, did anyone actually try any other programs other than CC Talk on your lab? I know someone actually messaged me to ask if that's okay. No? Okay, I can't remember who it was who messaged me, but I think there were actually people who tried different programs to actually determine the guide RNA software. So in case you guys didn't actually click on the other links, this is what the output or the input window actually looks like. So you can actually, again, if you look at the table on the right, yes, so on the right, you'll see that one actually lists the different campsites and the different organisms that you can use. So if you don't have a preference, you obviously don't need to use something as complicated as this. The output of it is also a little different from what you guys saw in your lab. So this one, it actually already tells you where the position is and what the possible mismatch is, and also a score. This one is specifically for activation and repression mostly. It's called CRISPR era. We don't use it too much. This one you guys should be extremely familiar with, CC Talk. And you guys should have seen something like this in your lab report. If you guys don't have this in your lab report, now would be a good time to panic. Alright, next we also have Chop. Again, they are all just very different softwares. The output for this looks like this. So hopefully, if as you guys see all this, you can already sort of gauge in the future if you work with CRISPR, which one might be easier to work with. Alright, so in your lab, you identify your guide RNA and your vector. But if you actually do want to work with it, you actually have to assemble both those pieces together. You actually have to do cloning. So in this case, how do you actually clone both those pieces together? This one is also related to what you guys did for your lab. So in the lab, I think the question was how do you identify something that cuts? So that's to actually create a knockout. But if you wanted to do any of the other functions mentioned just a little bit earlier today, such as editing, repression, or activation, there are different sort of Cas9 or different sort of systems that are more suitable for it. So for example, if you want to do something like an edit, you have to make sure it's a base editor, so something that's able to change an A into a C, and so on. Right. So the next couple of slides from now until the end are just all the examples of the different CRISPR-Cas systems out there. So this is your very generic CRISPR-Cas9 system. We like to say this is very 2012. People don't use this so much anymore, but it does work really well if you want to knock out your gene of interest. As far as CRISPR requirement goes, something that I mentioned earlier is you do need that campsite, that sort of stop signal for your CRISPR-Cas system to know where it can bind to. Other types of CRISPR systems that's out there is using your CRISPR-Cas9 system as well. If you actually introduce a donor sequence and ensure that your donor sequence has homologous regions with the area that you want to target, then it's actually able to have homologal recombination that can actually occur and it will take out your donor sequence. Does anyone need me to repeat that? Okay, I'll repeat that again. So how you make precise modifications on knock-ins is you still use your CRISPR-Cas9 system. So CRISPR-Cas9 would usually create a cut at your gene of interest. Everyone can follow that so far? Yes, cool. So when you have a cut, usually your cell just wants to repair it very quickly. But if you introduce a donor sequence, so say this is the cut, and you introduce a donor sequence where it has a homologous region, it has similar regions to what sort of flanking the cut site, then when you have homologous recombination happen, it can actually take out the entire sequence that's on the donor template. Okay. Everyone okay with that explanation? Okay, cool, right. So that's how knock-ins happen, and you can actually make edits that way as well. And it can be from as small as one base pair to as large as 2,000 bases so far. Technology is improving, so I'm sure one day you will hear something like your donor template is 10,000 bases, and that would be okay as well. Other systems that are out there is known as the multiplex system. This one is, they're all under a single promoter, but you can actually knock out or knock in 7 different genes at the same time. I think the most number of genes modified by a certain scientist was actually 52 genes at one time, which was a lot, and he did it in animals, which was pretty cool. Other uses of CRISPR or other systems of CRISPR is known as the Gecko system. Gecko system refers to specifically humans and mice, where people have already knocked out every single possible gene in the entire human and mouse genome. So they mostly use this to study things like cell viability, or what happens when you lose the function of a gene. So in Malaysia, Cancer Research Malaysia actually uses this quite a lot. And they use this because you do see a lot of genes that are dysmorphic or that are affected because of cancer. Likewise, when you are doing something like chemo, you also see disruption of genes. So they use this quite a lot to identify the success of the drugs that they are using. There's also anti-CRISPR. Anti-CRISPR is an inducible system primarily used in non-human systems. A lot of it is used in plants. And how it works is there's usually a photosensitive gene that's attached to it. So it's a lot like saying, if you turn on the light, you can turn on your CRISPR, and if you turn off the light, you can turn off your CRISPR function. So it's actually great for plants. So imagine if you are able to control something like making a plant more drought resistant, or making a plant be able to grow better in the rainy season. So you can use things like an inducible system for that. Great. So going back to one of the points that we talked about earlier, whenever we talk about genome editing, delivery plays a huge role. And delivery, if you guys remember when we talked about different viral vectors have a different size capacity. So if you are trying to introduce a very large gene, or even your CRISPR, say you're trying to target 52 different genes at the same time, that will probably mean your construct is very big, and you wouldn't be able to use maybe say AAV for that. So if you have a smaller CRISPR construct, in this case, if you have something like Staph aureus, Cas9, that's pretty small, so you can easily package in any of your viral vectors. Great. There's also your activation and repression CRISPR systems. These are known as CRISPR-I, CRISPR-A. So CRISPR-I stands for CRISPR interference, CRISPR-A stands for CRISPR activation. And what they've actually done with these CRISPR systems is they've actually removed that sort of scissors portion, if you guys remember from one of the earlier slides, because again, these are not permanent, so you don't want to cause any double-stranded breaks to actually remove the function of the gene. You usually just want to sort of almost like put something there to increase the transcription or reduce the transcription. So in this case, again, that's known as CRISPR-I and CRISPR-A. Very important to point out that it is reversible, so it doesn't permanently modify the genomic claim. When we talked about different transgenic animals as well, something that we saw that really sort of affected a lot of the older traditional methods is your epigenetic markers, and that's something that usually is not passed down. So if you are doing something like that, and you want to make sure that your genetic markers are similar to your parental strain, you can actually use CRISPR for that purpose. For example, the P300 system. So you can actually modify things like methylation, acetylation, and so on. And in this case, when you do modify your genetic markers, it can be inherited by your daughter cells. Okay, everyone okay so far? Okay, about 20 more slides ago. Alright, we also have CRISPR-based editing, also known as repair. And in this case, you have your cytosine-based editor, so you can convert a C to U. So in this case, this is usually done when you have, say, SNPs or polymorphisms that give your, like, say, plant A a certain advantage. So if you know that what you have is a U in plant A that makes it grow better, then you can actually use your cytosine-based editor to convert a C to a U to make sure that plant B also grows as well as plant A. There's also editing-based editor, and this one it converts an A to a G. And it's not just for plants. Say we talked about viruses, where we know that if there's a mutation, it makes it more transmissible. So you can actually try to use this to make it less transmissible. There's also the CRISPR rainbow system. As the name suggests, it's called CRISPR rainbow because it actually has sort of fluorochromes or fluorescent proteins tagged with up to 7 different colors. So this is used quite a lot in studies where if you want to study, say, the metabolic pathway, say you wanted to do something related to synthetic biology, and you don't know which genes are important, and you're trying to decide between the 7 genes, you can actually just use CRISPR to tag those 7 genes and sort of monitor the progression of where those 7 genes go to, the 7 proteins go to try to use that to maybe come up with a better genome or come up with a better synthetic gene. Another CRISPR system is called capture. In this case, it's to purify genomic regions. This is used quite a lot for novel proteins. So say you don't know, or say you've discovered a new protein and you don't know what it does, you can actually use this. And how it works is that it's an particle tag. So if you have a tag into a protein, then you can sort of use that information then to find out more about your novel protein. Alright, any questions before we move on to all the RNA systems? Right. So moving on to RNA systems, in the past we always talk a lot about how CRISPR is used for DNA targeting. So before this, whenever we talk about RNA modification, people often use RNAi. But since then, in the past couple of years, it's actually been modified to target RNA as well. So there's Cas13. So Cas13 in bacteria actually cleaves non-specifically. So it's non-specific because all the CRISPR systems actually originated from bacteria as an adaptive immune system. So that's why in bacteria itself, it actually just cleaves anything that looks like it's potentially a viral pathogen against the bacteria. But in non-bacterial systems, so if you're targeting, say, mammals or plants or animals, you can actually use it to target your RNA specifically. This is also pretty cool. This is known as the Sherlock system. So it still uses Cas13, which was seen in the slide before. And in this case, it is used to detect different nucleic acids at a very, very low level. So it actually has been used to differentiate strains of previously between dengue and Zika at the time when people did not know which illness or which virus they're actually dealing with, to genotype DNA and identify tumor mutations. So it's actually quite cool. How Sherlock works is a lot of times they actually put it on Sugar was actually also used as a detection method for COVID, though it's a natural flow method. Right, this is the last CRISPR system. So there's also CRISPR RNA-based editing, which is Cas13A, Cas13B. So quite similar to CDE and ABE that we saw earlier. In this case, you can convert an A to an I for the RNA-based editing system. Right, so this is the comparison between all the different CRISPRs that we saw earlier, the three main categories. So there's CRISPR9, so that's the one that we have seen over and over again, primarily used for knockout of proteins or genes. There's CRISPR12A, so this was the one where it was a bit smaller for easier delivery, and there's Cas13, which is primarily for RNA use. Alright, everyone still good? 16 more slides to go, two more main topics. So next is, say you've actually done all this, then you want to deliver it to your to yourself, to this organism of interest. This is mostly focused on plants or animals. We did touch a lot already on humans in some of the other slides, but also maybe right. I hope your laptop survived as well. Right. If not, then we can use Anpao to buy a laptop. Right. So for plants, you can use photographs transformation and work transformation combined with microparticles. For those of you who have probably already worked with plants, hopefully this is something that you're used to. For animals, you can use microinjection, evaporation, cryopreservation, or embryo transfer. We've also seen a lot of these techniques when we were talking about transgenic animals earlier in this semester. Right, this is the last part of the lecture today. So, say you've done everything. So you've identified a gene, you've cloned your CRISPR protein, you've delivered it to a cell of interest. How do you know if it actually works? So the validation component or the validation portion is actually also extremely important for you to verify the success of your experiment. So these are sort of the four main areas that people typically do. So in the early days, people used to do this thing called a mismatch, to which I say I'll tell you guys how that works shortly. A restriction digest to actually also verify whether it worked. Let me not show you how that works shortly. PCR amplification or sequencing. Alright, so these are the different validations that can be used. Some of it may be familiar to you, some of it may be quite new. So mutation detection is something that might be quite new. I'll talk about that in a bit. Fluorescence microscopy, flow cytometry, qPCR is something that is already typically used for a lot of cell and protein and RNA work. And same goes to Western blot and ELISA, that was used a lot in protein work. So you guys may not have heard of ddPCR, traps, and sequencing too much, but I'll talk about how that's used in CRISPR work. Alright, so at the very beginning, in the early days of CRISPR work, not so much anymore, a lot of people used to use mutation detection, and this was mostly used when they were trying to identify if a gene had been knocked out. So what they would do is, if you remember, once you have a double-stranded break, your cell tries to repair it. And it tries to repair it through a very sloppy NHDJ mechanism. So you have random insertions and deletions at the cut site. Not all cells will have the same insertion and deletion. So maybe one cell will have one insertion, another cell will have two deletions, another cell will have three deletions, and so on. So that will actually make it very hard to sequence, because if you do sequence something like that, what you get is just a lot of noise, because sequencing does require a certain threshold to be able to do clean sequences. So what people then did instead is this thing called mutation detection. So how that works is you would PCR amplify out that whole region. So about 1,000 pieces flanking your cut site. So when you do that, and then you actually denature it, what you would get is you get some strands where it's a bit shorter because of the deletion, and you have some strands which is a bit longer because of the insertion. And then after the denaturation step, they re-anneal it, and when you re-anneal it, because of that some shorter regions, some longer regions, you actually have mismatches. Can everyone sort of imagine that right now? Yes. Okay, cool. So because of the mismatches, they will then introduce an enzyme, T7, internuclease 1 or cell 1 enzyme, and this enzyme is actually able to detect where the mismatches are. So once it detects the mismatches, it actually causes a cut, and then if you run it on a gel, if you see multiple bands on the gel, it actually tells you that there are mismatches, which means that you have a double-stranded break, which means that your CRISPR worked. Okay, everyone okay? Okay, cool. So this is a very old method. Obviously, even hearing about it now, it sounds a bit complex, it sounds a bit long, and probably there are other better ways of actually detecting whether your CRISPR worked or not. Right. The other way for detection or validation is fluorescence microscopy. This can be done in several ways, especially if your CRISPR itself has GFP in it. So if your CRISPR itself has GFP in it, if you did actually successfully introduce your CRISPR plasmid into your cells, you would see higher expression of GFP as opposed to if you didn't. Same goes to flow cytometry, especially if you're using GFP. Actually, does everyone here know what flow cytometry is? It's what? So you can use flow cytometry to detect certain markers within cells. You can also detect for size, you can also detect for live plate cells, and so on. So in this case, if you are detecting GFP specifically, you're actually able to look or see something like this, where you have upregulation of GFP in something which is still work successfully, and you don't have GFP when you didn't introduce your CRISPR. You can also use qPCR. This is just a simple quantification of your RNA. So if you are knocking down your DNA, you should also see a decrease in your RNA as well. Likewise in your protein as well. So if you want to see whether you really do see a change in your transcription, you can just measure your RNA of what you initially targeted. ddPCR is something like qPCR, but it's about 20,000 times more sensitive. So in some cases, if you only have a success or a decrease of a certain expression by 0.01%, that may be too small enough to be able to detect through qPCR or other methods. So people like using ddPCR, which is a bit more sensitive in comparison to qPCR. For protein expression, you can use ELISA. Again, if you see a decrease in your DNA, you should also see that in your RNA and protein. This is typically used to detect certain cytokines and proteins. CAPS is if you had designed your CRISPR to disrupt a particular restriction enzyme site. So when you were looking at plasmids, you saw a lot of restriction enzyme sites, but it's the same in your genome as well. So if you know, for example, there's a certain MAH1 site in the region that you want to target, if your CRISPR did actually cut and introduce random insertions and deletions, the MAH1 site technically shouldn't be there. So you can actually use that to determine if your CRISPR worked or not. You can also use sequencing. This is especially used if you're trying to introduce certain genomic region, so knock-in, or if you are trying to make a certain base edit. So if you look at the top, you will see that your chromatogram has multiple peaks in the region which is highlighted in blue. In that case, this experiment was to actually make base edits. So when you see multiple peaks, it does tell you that you did get a successful, or you did successfully change your bases for the top. In the bottom one, it didn't work, so you actually just see your original sequences, and very easy, very clear, simple chromatogram. Everyone okay still? Cool. Alright, there are also other sorts of online software out there. This is called Indel Reconvolution. So what this program actually does is it sort of takes that chromatogram information and breaks it down into sequences for you. So remember, you will get something like a mixture of different insertions and deletions. So it actually breaks it down and tells you things like out of everything you sent in, you do see about half of it had a huge number of deletions, and another half had a huge number of insertions. So it does tell you that the CRISPR works well. Alright, so in summary, please ignore the last line. In summary, you can use all these different methods to detect all those different things out there or changes in your genome or genome of interest. And just to remind everyone, I guess you will see this in your lab talk again as well. But this is the workflow summary of CRISPR work from beginning to end. You start with your genome targets, designing your guide RNA, assembling a construct, delivering it to organism, and screening for gene editing. If you're interested in CRISPR work, this whole process takes roughly less than 2 weeks. Alright, just a simple quote from the person one of the main people who discovered CRISPR. She said that plants that can battle the climate change, mosquitoes that are unable to transmit malaria, these organisms may seem far-fetched, but they already exist thanks to gene editing, and they are only the beginning. As I write this, the world around is being revolutionized by CRISPR, whether we are ready for it or not. So, as usual, take-home points. Is genetic engineering more suitable for gene therapy, so for humans, or to make transgenic plants and animals? Which was the most important discovery that led to the success of genetic engineering? We saw things like insulin and other types of gene splicing and whatnot. Do you think that's the most important things? What are some other examples of crop and livestock modification? Can you describe the workflow of CRISPR? And will you be able to describe the differences between the Cas proteins? Alright, I think that's it for today. So I'll take any questions. Otherwise, if you guys don't mind passing me the names for your lab